Assalamualaikum, I'm Umi Sumaya binti Hamdi. My student ID is 2023462842. So now I want to talk about the introduction of thin layer chromatography, which is TLC, in forensic analysis. Okay, so chromatography is a technique used to separate and identify components in a mixture. Thin layer chromatography or TLC is a quick and cost-effective method that uses a thin silica gel layer as the stationary phase and a mobile phase to move the sample components at different rates. In forensic science, TLC is very useful. For example, by analyzing pigments in different lipstick brands, we can identify similarities or differences which may help in criminal investigations like theft or assault. This experiment demonstrates how TLC separates chemical components and shows its practical application in real-world forensic analysis. Using this method, we can compare lipstick samples quickly and visually, linking lab techniques with actual investigative work.
Hi, Assalamualaikum. My name is Nafis Nabila Khairnizam. My student ID is 2024-902-625. Today, I will be presenting about theory. In this experiment, TLC is used to compare the red pigment present in different lipstick samples. Lipsticks are complex mixture that may contain several dyes and pigments. In TLC, a small amount of lipstick spotted onto a TLC plate is dissolved in a solvent with a stationary phase such as silica gel or alumina. Then the plate is placed in a developing chamber containing a mobile phase. As the solvent rises up the plate, it carries the pigment molecules with it. The separation occurs because different pigment interact differently with the stationary and mobile phase. More polar pigment tend to bind more strongly to the stationary phase, therefore travel a shorter distance up the plate. More polar pigment interact more with the mobile phase and move further up the plate. After development, the separated pigment become visible either naturally due to their color or under UV light. The distance traveled by each pigment spot is measured and compared to the distance traveled by solvent front to calculate the retouch vector. Pigment with the same or similar RF values across different lipstick samples are likely to be the same compound, while the different RF value is a different pigment. By comparing the number of spots, the compound and the RF values, conclusion can be decided about similarities and differences in the red pigment used in various lipsticks.
Hi, Assalamualaikum. My name is Nur Shakira Shahmina binti Nur Amali. My student ID is 2024539225. Today, I will talk about the methodology of application of thin layer chromatography in a forensic analysis to comparing pigment composition across lipstick brands. Firstly, the sample preparation. For the methodology, several commercial lipsticks brands with similar red shades were selected to ensure that visual comparison alone was insufficient, similar to real forensic cases. Each lipstick sample was smeared onto filter paper to simulate forensic trace evidence. A small portion of the stained filter paper measuring 4 mm by 4 mm was then cut and placed into a test tube. Next, 1.0 mm of petroleum ether was added to remove waxes and oils present in the lipstick. This extraction process was repeated twice, and the petroleum ether was discarded after each extraction. After removing the waxy component, the coloring agents were extracted using a methanol ammonium hydroxide solution in a 95:5 ratio. The extract was gently heated to reduce its volumes, and finally 2 drops of methanol were added before spotting the sample onto the TLC plate. Next, the TLC analysis and comparison. For the TLC analysis, silica gel aluminium TLC plates with fluorescence indicator F254 were first activated at 100 degrees Celsius for 1 hour. After cooling, the sample were carefully spotted onto the baseline using a capillary tube. The mobile phase used was a mixture of chloroform, methanol, and water in the ratio of 50 to 15 to 2. The TLC plate was then developed until the solvent front reached approximately 3/4 of the plate's length. After development, the plate was air-dried and the separated pigment spots were observed under both white light and ultraviolet light. The RF value for each pigment band were calculated. Finally, the lipstick brand will compare based on the number of spots, the color of the pigment band, and their RF values to demonstrate the forensic application of TLC in different tinting lipstick samples.
Hello and Assalamualaikum. My name is Nurulajra binti Mazamri and my student ID is 2024959 and 795, and I will continue with the example of the results. Lipsticks or similar colors were selected. To simulate evidence, smears were rubbed onto a clean cotton cloth. These stains were then cut and transferred to a bowl, where they were mixed with an extracting solution, which is stored in for 5 to 10 minutes to remove the pigment from the fabric. A stationary phase, which is the silica gel, plate was prepared with a pencil line drawn 1.5 cm from the bottom. The microliters of the extracted lipstick were spotted on this line using capillaries. Then the plate was placed in a jar containing a mobile phase, which is the solvent system, such as toluene, benzene, acetone, and diethyl ether. Once the solvent reached near the top, the plate was removed. Since many pigments are faint, the plates were exposed to iodine fumes to make the spots clearly visible. Researchers then measured the distance traveled by each component to calculate the HRF value. The red dye concentration cannot identify specific lipstick brands because the data isn't unique enough to tell them apart. Even though pigment level vary across all samples, this difference happened in both local and well-known brands. The main finding was that the local lipsticks are far more complex, usually containing more than 2 dyes, while the branded one usually have 1 or 2. However, because of their HRF value are so similar, there is no special features to distinguish one brand from another. In conclusion, this study shows that the thin layer chromatography is a cheap and useful way to test lipstick found at the crime scenes. Since lipstick is the most important evidence, this test helps to connect a person to a crime. This study found that local lipsticks are far more complex because they usually have more than 2 dyes, while big brands usually have 1 or 2. However, the test has limits. It is hard to tell exactly which brand is which because most lipsticks use similar red dyes that look the same in the result. Because of this, TLC is a great extra tool for looking at pigments, but it cannot identify a specific brand all by itself.